There is a moment in the history of life on Earth that scientists have been trying to explain for more than 150 years and have still not fully explained. A moment where something changed so radically and so rapidly that the rocks recording it look like a before and an after with no visible transition. A moment that Charles Darwin recognized as the greatest problem for his theory of evolution, and that today, with all the instruments of molecular biology, paleontology, and geochemistry, remains one of the most debated and most fascinating events in the entire history of life. The Cambrian Explosion is also a story about scale that is difficult to hold in mind. Not just temporal scale, though 540 million years is difficult enough to comprehend. But biological scale, the difference between a world with no complex animals anywhere on the planet and a world suddenly full of them. The Ediacaran Ocean, for all its strange organisms, was a world where if you could somehow travel through it for millions of years, you would never encounter anything that hunted you, that saw you, that had the biological machinery to recognize you as prey. And then, in the Cambrian, that changed everywhere at once. The beauty of the Cambrian also lies in what it tells us about the relationship between simplicity and complexity. For 3 billion years, life on Earth was microbial, simple, invisible to the naked eye. And then, in what seems like an instant in geological terms, complexity exploded. Eyes appeared. Predators appeared. Shells and armor appeared. That transition from bacterial world to animal world, from microbial ocean to an ocean full of creatures with behaviors and interactions, is one of the most dramatic phase transitions in the history of nature. The Cambrian is the moment the ocean became interesting in the way that oceans are interesting today. Tonight, as sleep approaches, the Cambrian Ocean is still here in the sense that matters most, in the biological inheritance it left behind. In the eyes reading these words, in the spinal cord carrying the signal of each breath, in the genome encoding the developmental programs that built every structure of the body now resting. 540 million years ago, an ocean filled with life for the first time. Tonight, that life rests. The ocean of the Cambrian also had its own music. Not the music humans produce with instruments, but the vibration of water, the movement of organisms, the currents circulating through ancient seas. In that ocean without vertebrates with jaws and without the vocal structures vertebrates would later develop, the sounds were those of shells clicking, water displaced by swimming bodies, and the deep silence of the darker waters where sunlight did not reach. That moment is the Cambrian Explosion. 540 million years ago, the ocean changed. Not gradually, not over the course of hundreds of millions of years. In the space of a few million years, a blink in the scale of Earth's history, the ocean that before was dominated by simple soft-bodied organisms filled with animals that had eyes, mouths, claws, shells, armor, and legs. Animals that hunted other animals. Animals that built structures to protect themselves. Animals that saw, that swam actively, that burrowed into sediment searching for food or shelter. Before the Cambrian Explosion, complex life was scarce and mysterious. The organisms of the previous period, the Ediacaran, were strange-looking creatures that paleontologists still debate whether they were animals, plants, fungi, or something completely different from any category we use today. Some resembled leaves, others discs, others tubes. None had eyes. None had a mouth in the sense we understand a mouth. None had hard parts protecting them from predators, probably because there were no predators. And then, in the Cambrian, all of that changed at once. Animals appeared with all their complexity, with their vision systems, their digestive systems, their defenses. The world went from being an ocean of soft, enigmatic creatures to being an ocean of predators and prey, of evolutionary arms races, of life in all its violent and beautiful complexity. The Cambrian Explosion is also, in a very deep sense, the story of how the world learned to die in new ways. Before the Cambrian, organisms died by accident or by adverse environmental conditions, but rarely died because another organism actively killed them. In the Cambrian, active predation became a dominant evolutionary force, and death by predation drove evolution in a way that no abiotic factor could match. The life we know today, all its complexity and all its diversity, is largely the result of that Cambrian invention, the arms race between hunter and hunted. Tonight we are going to explore that world with the slowness it deserves, with the attention those impossible creatures require. We are going to swim with the Anomalocaris and walk the seafloor with the Hallucigenia. We are going to understand why that particular moment, in that particular ocean, was the one that changed the history of life forever. To understand the Cambrian explosion, one must understand what came before. The Ediacaran, the period immediately preceding the Cambrian, lasted from approximately 635 million years ago until 541 million years ago, almost 100 million years of a world radically different from what followed. The trace fossil record of the late Ediacaran, just before the Cambrian, shows a progressive increase in the complexity and diversity of movement traces. The first Ediacaran organisms left simple horizontal movement traces on microbial mats, as if gliding across the sediment surface. Late Ediacaran traces are more complex, with evidence of vertical burrowing into sediment and more active movements. That progression in trace complexity is one of the most direct pieces of evidence that diversification of animal behaviors began before the formal Cambrian boundary, though it was in the Cambrian where it reached maximum expression. The Ediacaran organisms that survived longest into the Cambrian provide some of the most interesting evidence about the nature of the transition between the two periods. Some Ediacaran forms persisted for millions of years into the early Cambrian before finally disappearing, coexisting with the first shelled animals and the earliest trilobites. That period of coexistence between Ediacaran and Cambrian organisms is recorded in some rock sequences with enough detail to allow studying whether the two groups interacted directly or simply occupied different parts of the available ecological space. The question of whether the Ediacaran organisms were truly animals or something else entirely is more than a taxonomic curiosity. If they were animals, then the Cambrian did not invent animal life but accelerated it. If they were a separate kingdom of life that went extinct completely, then the Cambrian truly invented animals from scratch. Most researchers today lean toward intermediate positions. Some Ediacaran organisms were likely early animals, while others may have been something genuinely different from any modern category. The Ediacaran-Cambrian boundary is also one of the few geological boundaries visible to the naked eye in many rock outcrops. The change from the thin laminated Ediacaran sediments, with their characteristic microbial mat textures, to the bioturbated Cambrian sediments is often abrupt and dramatic in hand specimen. Geologists who work in those transitions describe it as a fundamentally different-looking rock record, as if the world that produced the sediment had changed completely. That visual change in the rock encapsulates the biological revolution that occurred at that boundary in a form anyone can observe. The Cambrian explosion is sometimes discussed as if it were a single sudden event, but it was actually a process unfolding over millions of years, with different groups diversifying at different times. The first mineralized shells appear approximately 541 million years ago. The first trilobites appear approximately 520 million years ago. The Burgess Shale community, one of the most diverse known Cambrian assemblages, dates to approximately 508 million years ago. That temporal spread within the Cambrian means the explosion was a rapid but not instantaneous process. The Ediacaran is in many respects the most mysterious period in the history of complex life. Its organisms, collectively known as the Ediacara biota after the Australian sites where they were first described in the 1950s, are creatures of a strangeness that defies classification. Dickinsonia, one of the most common and most studied Ediacaran organisms, was a flat organism with an oval leaf-like appearance marked by segments that could reach 1.5 meters in length. Charnea was a branching form similar to a feather that attached to the substrate at one end. Ediacaria was a disc living in the sediment whose exact function is still debated. The Ediacara biota also included organisms recently interpreted as the first animals with confirmed bilateral symmetry. The Icaria wariusha, described in 2020 from small fossil traces in Australia, is considered the oldest known bilaterian, approximately 570 million years old. Although much smaller and simpler than Cambrian animals, its bilateral symmetry, with a clearly differentiated front and back, is the same symmetry characterizing all modern vertebrates, arthropods, mollusks, and worms. The transition from Ediacaran to Cambrian was not only biological. It was also geological and geochemical. The rocks marking the boundary between the two periods record changes in the isotopic composition of carbon and strontium, reflecting deep changes in ocean chemistry and the global carbon cycle. Those geochemical changes coincide with the biological changes documented in fossils, suggesting the transition to the Cambrian was accompanied, and probably facilitated, by changes in ocean conditions that made possible or favored the diversification of complex animals. The extinction of the Ediacara biota is another fascinating aspect of the transition. Ediacaran organisms do not disappear from the fossil record either gradually or in an instantaneous catastrophe. They disappear progressively through the early Cambrian, while the first Cambrian animals diversify. Some researchers argue that the appearance of the first animals with mouths in the early Cambrian was the direct cause of Ediacaran extinction. The first Cambrian predators and burrowers disrupted the microbial mats on which Ediacaran organisms depended and converted the sediment from a stable substrate into a constantly disturbed environment. The Cambrian Ocean was a physically different place from the modern ocean, not only in terms of the organisms inhabiting it, but in terms of its chemistry, its geography, and the conditions determining what could live in it. The geography of the Cambrian was completely different from today's. None of the continents we know today existed. The Cambrian continental blocks were land masses of completely different configuration, distributed in positions that only make sense when one knows the history of continental drift. The supercontinent Gondwana already existed, but in a different position. The future continent of Laurentia, which would eventually form the core of North America, was a separate landmass in tropical latitudes. The ocean covering most of the planet, the Panthalassa, was an immense global ocean with no equivalent in the modern world. The chemistry of Cambrian ocean water also differed in important ways that influenced what organisms could build and how they built it. The calcium carbonate saturation of the Cambrian Ocean was higher than today, meaning that calcium carbonate shells and skeletons were thermodynamically favored to form and relatively resistant to dissolution. That chemical environment was one of the factors facilitating the proliferation of calcite-shelled organisms in the Cambrian. When ocean chemistry changed in later periods and calcite became less stable, different mineralogies became favored for shell construction, and the composition of marine fauna reflected those chemical changes. The Cambrian seafloor was not uniform. Like modern ocean floors, it was topographically varied, with shallow banks, channels, deeper basins, and the occasional seamount or volcanic structure. Those variations in topography created variations in current patterns, sediment deposition, oxygen levels, and food availability that would have driven ecological differentiation among the Cambrian organisms inhabiting different parts of the seafloor. The spatial variation in Cambrian communities that paleontologists observe in different parts of the same geological formations reflects that underlying topographic and environmental heterogeneity. The Cambrian Ocean was also characterized by the absence of things we take for granted in modern marine ecosystems. There were no grasses, no kelp forests, no mangroves, no large marine plants of any kind. The photosynthetic base of the food chain was entirely planktonic, consisting of algae and cyanobacteria floating in the water column. That difference in the structure of primary production had consequences for how energy and nutrients flowed through the Cambrian ecosystem compared to modern marine ecosystems. The Cambrian seas also lacked fish. That might seem obvious given that fish had not yet evolved, but its ecological implications are profound. The modern ocean is structured in large part around the activities of fish. Their predation, their excretion, their migration, their decomposition after death. Without fish, the Cambrian Ocean's nutrient cycling, predator-prey dynamics, and water column structure were all fundamentally different. The world before fish is almost as hard to imagine as the world before animals, yet that is what the Cambrian Ocean was for most of its duration. The Cambrian also lacked the large land plants that today intercept rainfall and regulate the flow of nutrients from land to sea. In the Cambrian, rain fell directly on bare rock, eroding it efficiently and delivering large quantities of dissolved minerals to the ocean. That higher mineral flux from the continents contributed to the elevated calcium and nutrient concentrations in the Cambrian Ocean that supported the explosion of complex life in its waters. Sea level during the Cambrian was significantly higher than today. The combination of a planet without permanent polar ice caps and younger and therefore less dense oceanic lithosphere produced a global sea level that flooded extensive areas of the continents, creating broad, shallow epicontinental seas. Those warm, shallow seas, with carbonate floors and transparent waters allowing sunlight to penetrate to the bottom in the shallowest zones, were the richest habitats of the Cambrian. The sedimentology of the Cambrian also records the first events of massive bioturbation in history, when Cambrian organisms began burrowing into sediment and disturbing it in ways that transformed its structure. Before the Cambrian, marine sediments showed perfect fine lamination because there were no organisms disturbing them. In the Cambrian, that lamination was progressively destroyed by bioturbation, and late Cambrian sediments are much more disturbed than early Cambrian ones, reflecting the diversification of burrowing organisms through the period. The temperature of the water in the Cambrian Ocean varied with depth in a way similar to modern oceans, though gradients may have differed. Warm surface waters, sunlit and productive, contrasted with the cooler, less oxygenated deeper waters. That thermal and chemical stratification of the ocean created different inhabitable zones with very different conditions, and Cambrian organisms were beginning to adapt to each of those zones in specific ways. The Cambrian oceanic circulation was also different from today's. Without the great continental masses that today direct ocean currents, the Panthalassa could circulate more freely around the planet. That freer circulation could have distributed heat more uniformly between the tropics and poles, contributing to the warmer global climate of the Cambrian, and could have distributed nutrients more efficiently, supporting the primary productivity that sustained the food chains of Cambrian ecosystems. In the Cambrian Ocean, there was an animal that was the greatest predator of its time, the largest organism and probably the most formidable in an ecosystem full of creatures that for the first time in life's history were learning to hunt. It was called Anomalocaris, which in Latin means anomalous shrimp, and it is one of the most extraordinary animals that has ever existed. The Anomalocaris and its radiodont relatives also illustrate an important principle about the evolution of ecological complexity, that the appearance of a new ecological role, like apex predator in a complex food web, can drive the diversification of all other roles in that food web. Once Anomalocaris appeared and began selecting for well-defended prey, every lineage of Cambrian animals faced new pressures that had not existed before. Those pressures drove diversification not just in potential prey but in every organism whose ecology was affected by the changed competitive and predatory landscape. The predatory innovations of the Cambrian were not limited to the large active predators like Anomalocaris. Some of the most ecologically important predatory innovations were at smaller scales: the evolution of jaws-like structures that could pierce and grasp, the evolution of venoms and chemical defenses, the evolution of ambush strategies using camouflage, and the evolution of speed as a prey escape mechanism. All of those smaller-scale evolutionary responses to the predation arms race collectively transformed the Cambrian ecosystem into something far more complex and dynamic than it had been in the Ediacaran point. One of the most striking aspects of Anomalocaris and the other large Cambrian predators is how quickly they appeared in the fossil record. Within a few million years of the first appearance of complex animals, apex predators capable of hunting large prey were already present. That rapid evolution of complex predatory behaviors and morphologies from simpler ancestors is one of the most direct demonstrations of how powerful selective pressure from ecological interactions can be in driving evolutionary change. The Anomalocaris also illustrates the way the Cambrian produced ecological roles before the organisms filling them had completely stabilized. As an apex predator in a world where all other ecological roles were still being established, Anomalocaris was shaping an ecosystem that was simultaneously shaping it. The prey it hunted were evolving harder shells in response to its predation. Those harder shells in turn selected for stronger grasping appendages and more powerful bite forces in Anomalocaris. That coevolutionary feedback loop was itself a novelty of the Cambrian, the first evolutionary arms race in the history of complex life. The Cambrian radiodont diversity also shows how a single successful body plan can be modified into many ecological variants. The basic radiodont body plan, with frontal appendages, a circular or oval mouth, and a streamlined body with lateral swimming structures, was elaborated into active predators, filter feeders, bottom skimmers, and possibly other ecological variants. That radiation from a single body plan into multiple ecological roles within the same geological period is a pattern repeated throughout evolutionary history. Anomalocaris had a soft flattened body up to 1 meter in length, with undulating lateral fins allowing it to swim with efficiency and precision. At the front of its body it had 2 large articulated grasping appendages covered in spines, which it used to capture prey. And at the center of those appendages it had a circular mouth with toothed plates arranged like a pineapple, capable of opening and closing to crush even the hardest shells. The biomechanics of Anomalocaris, studied using computational fluid dynamics models simulating its movement in water, reveal it was an efficient swimmer capable of sustained speeds and precise maneuvering. Its undulating lateral fins, similar in principle to those of modern squid and cuttlefish, provided smooth and silent propulsion, ideal for stalking prey without producing water disturbances that might alert them. The combination of speed, maneuverability, excellent vision, and efficient capture appendages made Anomalocaris a truly formidable predator in the Cambrian ecosystem. What makes the history of Anomalocaris especially fascinating from a paleontological perspective is that for decades nobody knew what it was. The different body parts were described as 3 different animals. The grasping appendages were described as headless shrimp, hence the name. The circular mouth, when found as an isolated fossil, was described as a jellyfish. And the rest of the body was described as a sponge. Only when paleontologists at the Burgess Shale in Canada found complete and well-preserved specimens in the 1980s did they understand all those pieces belonged to a single extraordinary animal. The traces Anomalocaris left in the fossil record are not only its own fossils. They are also the marks left on prey. In some Cambrian sites, trilobite shells preserve bite marks with a specific pattern that exactly matches the geometry of the Anomalocaris circular mouth. Those bite marks are direct evidence that Anomalocaris actively attacked trilobites, and the fact that some trilobites survived those attacks, showing shells that healed around the wound, indicates those encounters were not always fatal. Anomalocaris belongs to a group called radiodonts, primitive arthropods with specialized frontal appendages that diversified in the Cambrian into multiple forms and sizes. Some Cambrian radiodonts were active predators like Anomalocaris. Others were filter feeders, with comb-like appendages filtering water for small organisms. That functional diversity within a single primitive group is typical of the Cambrian, where new body plans were exploring an ecological possibility space that no one had occupied before. If Anomalocaris was the dominant predator of the Cambrian, trilobites were the dominant animals in terms of abundance and diversity. Trilobites are the most iconic fossils of the Paleozoic, their articulated shells preserved in millions of specimens in the rocks of the Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian. For almost 290 million years, they were one of the most successful groups in the history of animals with skeletons. The history of trilobites illustrates another important principle of evolutionary biology, that success in one geological period does not guarantee survival through geological crises. Trilobites were extraordinarily diverse and abundant for almost 300 million years, surviving multiple mass extinctions that devastated other groups. Their segmented exoskeletons, multiple ecological adaptations, and global distribution made them one of the most resilient animal groups in Paleozoic history. And yet the Permian-Triassic extinction, the largest mass extinction in the history of complex life, eliminated them completely. The same properties that made them successful in normal times, particularly their dependence on the specific marine ecological conditions of the Paleozoic, made them vulnerable to the catastrophic changes of the Permian-Triassic boundary. The diversity of trilobite reproductive strategies also tells us something important about the ecological complexity of the Cambrian. Some trilobites produced large numbers of small eggs, suggesting a strategy emphasizing quantity over parental investment. Others show evidence of protecting eggs in a brood chamber under the cephalon, suggesting some form of parental care. And the presence of trilobite instars, the successively larger juvenile molts that reveal ontogenetic stages, in some fossil assemblages suggests that at least some Cambrian habitats were nursery grounds where juvenile trilobites were concentrated, perhaps for protection. The trilobites also provide some of the best evidence for the evolution of complex behaviors in the Cambrian fossil record. Some trilobite species are found in aggregations that may represent mating assemblages, similar to the spawning gatherings of modern horseshoe crabs, their distant relatives. Others show evidence of migratory behavior, with consistent orientations suggesting directional movement across the seafloor. Those behavioral complexities, preserved in the distribution and orientation of fossil specimens, hint at the richness of Cambrian animal behavior that the morphological record alone cannot reveal. The extraordinary abundance of trilobite fossils in Cambrian and subsequent Paleozoic rocks is partly a consequence of their molting behavior. Like all arthropods, trilobites shed their exoskeletons periodically as they grew. Each shed exoskeleton, called an exuvia, had the potential to be preserved as a fossil. A single trilobite over its lifetime might produce a dozen or more molts, each one potentially preserved in the rock. The result is that trilobite fossils are enormously more common than the actual number of trilobite animals would suggest, making them among the most abundant fossils in Paleozoic rocks worldwide. The trilobite fossil record is also one of the longest and most detailed records of evolutionary change in any animal group. Because trilobites were so abundant and so widely distributed, their fossils document evolutionary changes across geological time, geographic space, and ecological conditions in a way that few other fossil groups can match. The study of evolutionary change within trilobite lineages, using large samples of well-dated specimens, has produced some of the most rigorous tests of evolutionary hypotheses available in the paleontological literature. Trilobites were marine arthropods, distant relatives of modern crabs, spiders, and insects. Their most recognizable characteristic was their exoskeleton divided into 3 longitudinal lobes, hence their name. That exoskeleton was hard and calcified, making them extraordinarily good candidates for fossilization and explaining why their fossils are so abundant in Paleozoic rocks. The mineralogy of trilobite exoskeletons also provides information about Cambrian ocean conditions. Early Cambrian trilobites had exoskeletons with relatively high magnesium content, while late Cambrian ones had less magnesium. Those changes in the mineral composition of exoskeletons reflect changes in seawater chemistry through the Cambrian, which in turn reflect changes in the geological processes controlling ocean composition. The trilobites of the Cambrian already showed considerable ecological diversity. Some were active predators hunting worms and other soft invertebrates in the sediment. Others were detritivores moving across the seafloor eating decomposing organic matter. Some swam in open water. Others burrowed in the sediment. And many had the ability to roll into a ball when threatened, exposing only the hard shell and protecting the soft body parts beneath. Trilobite eyes are also extraordinarily interesting. Trilobites had compound eyes formed by calcite lenses, the same mineral forming marble and limestone, rather than the protein lenses of modern insect compound eyes. Those calcite lenses were rigid and perfectly transparent, making trilobite eyes high-quality optical systems. Analysis of trilobite eye fossils has revealed some had thousands of individual lenses and a nearly 360-degree visual field, making them difficult to surprise from any direction. Trilobites are also important for understanding Paleozoic paleogeography. Different trilobite groups inhabited different regions of the planet, separated by oceanic barriers preventing their dispersal. By studying the distribution of different trilobite genera and species in Cambrian rocks from different continents, paleontologists can reconstruct the relative positions of those continents during the Cambrian with precision other methods do not always allow. The Cambrian was not only the period of trilobites and Anomalocaris. It was also the period of some of the strangest creatures that have ever lived on this planet, organisms whose appearance is so unusual that paleontologists took decades to understand even what they were and how they lived. The Cambriaster, discovered relatively recently in the Burgess Shale, was a large radiodont with a head that has the shape surprisingly similar to the F-117 stealth fighter, which earned it the informal nickname spaceship among the paleontologists who described it. Its head shape suggests it was a sediment filter feeder that plunged its head into the seafloor to search for food in the substrate. That Cambrian organism thus occupied an ecological niche similar to that of certain modern rays and flatfish that also search for food buried in marine sediments, another example of ecological convergence across hundreds of millions of years. The extraordinary diversity of Cambrian body forms also raises the question of why similar body plans were not reinvented after the Cambrian. If natural selection is so powerful that it repeatedly finds similar solutions to similar problems, as shown by examples of convergent evolution throughout evolutionary history, why did it not produce new phyla in the hundreds of millions of years after the Cambrian? The answer probably involves the concept of evolutionary constraints. Once body plans are established with their complex gene regulatory networks, developmental pathways, and ecological specializations, modifying them radically is constrained in ways that the first Cambrian animals, with their less integrated developmental systems, were not. The Cambrian also produced some of the first examples of what biologists call convergent ecological interactions, situations where unrelated organisms evolved to play similar ecological roles in different times or places. The filter feeding strategy employed by Cambrian radiodonts like Peytoia, using comb-like appendages to strain small organisms from the water, was independently evolved by whale sharks, basking sharks, and manta rays in the modern ocean. That convergence across 500 million years of evolution demonstrates that filter feeding from open water is a stable ecological strategy that natural selection repeatedly discovers when the body plan and ecological conditions permit. The Cambrian's legacy in modern art and popular culture is also worth noting. The strange creatures of the Cambrian, particularly after Stephen Jay Gould's Wonderful Life brought them to public attention in 1989, have inspired artists, designers, and writers. The Anomalocaris has appeared in natural history documentaries, video games, and science fiction. The hallucigenia, with its initially puzzling orientation, became a symbol in popular science writing for the way scientific understanding can be wrong in systematic and amusing ways. The Cambrian has found a cultural presence that few geological periods can match. The Nectocaris was an animal that for decades was considered one of the most mysterious of the Burgess Shale, with a body not clearly resembling any known group. When new specimens were described in 2010, its reinterpretation as the most primitive known cephalopod was a surprise for the paleontological community. If that interpretation is correct, cephalopods, with their extraordinary intelligence and sophisticated adaptations, have their roots in that enigmatic Burgess Shale organism, connecting the Cambrian directly to the modern octopuses and squids that are considered among the most cognitively complex invertebrates alive today. Hallucigenia is perhaps the most famous of those strange organisms. It was a small creature, just a few centimeters in length, with a tubular body from which projected downward a series of rigid spine pairs and upward a series of soft tentacles. When first described from Burgess shale fossils, paleontologists interpreted it upside down. The spines, which actually served as support, were interpreted as dorsal tentacles, and the soft tentacles, which were actually walking appendages, were interpreted as a pair of ventral spines. Only when better preserved specimens were found in China, with head and tail clearly distinguishable, could the interpretation be corrected. Hallucigenia turned out to be a lobopod, a primitive ancestor of modern onychophorans, those velvet worms living in tropical humid forests that are the closest living relatives of arthropods. Seeing it correctly oriented, walking on its spines across the Cambrian ocean floor, does not make it much less strange. It remains a creature of remarkable alienness. The Cambrian was also the period when animals first learned to use color as a signal. Evidence of pigmentation in exceptionally preserved Cambrian fossils, though scarce and difficult to interpret, suggests some Cambrian organisms had colorations that could have served for recognition of conspecifics, for camouflage, or for warning predators they were venomous or difficult to digest. If those interpretations are correct, visual communication between animals, one of the most sophisticated forms of communication in the modern world, has its roots in the Cambrian. Opabinia was another extraordinary Cambrian animal. It had 5 eyes on the top of its head, a segmented body with lateral fins, and a long flexible proboscis with a claw at the end that it probably used to capture prey and bring it to the mouth. When presented at a scientific conference in 1972, the audience laughed when the reconstruction was shown. Not from malicious incredulity but from genuine astonishment at the animal's strangeness. Opabinia, with its 5 eyes and its claw at the end of a proboscis, seems drawn from a science fiction film, and yet it existed, swam in the Cambrian Ocean, and hunted its prey for millions of years. The discovery of new Cambrian species continues in known sites and in previously unexplored regions, reflecting how much remains to be learned about this fundamental period. Expeditions to the Canadian Arctic, the mountains of Morocco, the plateaus of China, and many other regions continue producing new fossils that expand and modify our picture of the Cambrian. Each new species adds a piece to the puzzle. Each new site provides a window into a Cambrian ecosystem in a different geographic context. One of the most important evolutionary innovations of the Cambrian, perhaps the most important of all, was the evolution of eyes. Not primitive eyes, which in the form of simple photosensitive patches already existed in earlier organisms. But true eyes, complex, capable of forming images, of distinguishing shapes and movements, of providing detailed spatial information about the environment. The evolution of eyes in the Cambrian also had consequences that go beyond predation and defense. Eyes allow navigation, spatial orientation, recognition of conspecifics, and potentially signaling between individuals of the same species. Those additional capabilities of eyes could have contributed to the evolution of social behaviors and more complex reproductive strategies in Cambrian animals. The importance of the Cambrian for understanding brain evolution specifically deserves emphasis. The vertebrate brain did not appear suddenly in the Cambrian. It was built gradually from the basic neural organization of the first chordates. But the first differentiation between a distinct anterior brain region and a posterior spinal cord, which is the defining characteristic of the vertebrate nervous system, appears to have occurred in the earliest vertebrates of the Cambrian. Understanding how and why that differentiation occurred is one of the central questions in the evolution of neuroscience, and the Cambrian fossil record, despite its incompleteness regarding soft tissue preservation of nervous tissue, provides essential context for those questions. The visual revolution of the Cambrian also transformed the ocean as a communication environment. Before eyes capable of forming images, signals between organisms were limited to chemical gradients, pressure waves, and other mechanisms effective only at short range or in specific conditions. With the appearance of image-forming eyes, The ocean became a medium through which information could be transmitted visually across distances of tens of meters, opening up new possibilities for signaling, recognition, and social interaction that had not previously existed. The modern ocean, with its extraordinary diversity of visual signals in fish coloration, cephalopod chromatophores, and crustacean displays, is the elaborated descendant of that visual revolution begun in the Cambrian. The relationship between the Cambrian visual revolution and the subsequent evolution of animal coloration is also worth considering. Once eyes capable of discriminating color evolved in the Cambrian, natural selection could act on the coloration of animals in new ways. Animals that could match their background became harder for predators with good vision to detect. Animals that could signal their toxicity or unpalatability through warning colors benefited from predators that could learn to avoid them. And animals that could produce specific color patterns recognized by conspecifics could use those patterns for mate choice and social communication. All of those color-based adaptations, which are so widespread in modern animals, were made possible by the visual revolution of the Cambrian. The behavior traces preserved in the Cambrian fossil record, in the form of locomotion and excavation traces in the sediment, show patterns that in some cases suggest coordinated behaviors between multiple individuals. Those evidences of gregarious behavior in Cambrian organisms are intriguing though difficult to interpret with certainty, but suggest that even in the Cambrian, the first complex animals were already developing forms of social interaction beyond simple predation and defense. Modern compound eyes can detect polarized light, which underwater is useful for navigation and communication because it creates patterns invisible to vertebrate eyes. Some arthropods use polarized light signals for mating displays and territorial marking. If Cambrian compound eyes had the same polarized light detection capability, the visual world of the Cambrian Ocean may have been far richer in structured information than we can fully appreciate from our vertebrate visual perspective. The appearance of eyes in the Cambrian was probably one of the most important drivers of the Cambrian explosion itself. The visual trigger hypothesis, proposed by paleontologist Andrew Parker in the early 21st century, argues that the evolution of image-forming eyes was the key event that unleashed the predator-prey arms race characterizing the Cambrian. When the first animals developed eyes capable of forming images, they could see their prey. Prey that could not be seen by predators did not need shells or spines for protection. But once predators could see, prey without protection were rapidly eliminated by natural selection, and those with some form of defense survived better. That selective pressure drove the evolution of shells, spines, and other defenses in prey, which in turn drove the evolution of more efficient jaws and appendages in predators. The genes PAX6 and their relatives controlling eye development in all animals are also a demonstration of the depth of the genetic heritage we share with Cambrian organisms. Those genes evolved in the Cambrian or before and have been conserved with remarkable similarity across all animal lineages with eyes. That a mouse eye development gene can function in a fly, producing a functional eye though morphologically different, illustrates that the basic molecular machinery of the eye was established in the Cambrian, and that same mechanism, with superficial modifications, still operates in every animal that sees today. The diversity of eye types in modern animals, with the compound eyes of arthropods, the lens eyes of vertebrates, the camera eyes of cephalopods, the mirror eyes of scallops, and other less common designs, reflects the radiation of solutions to the vision problem that occurred in the Cambrian. Each of those eye types has its own independent origin, its own evolutionary history, but all of them began in the same period, driven by the same selective pressures. That evolutionary convergence, multiple independent solutions to the same problem produced by the same pressures in the same period, is one of the most powerful demonstrations that evolution, though contingent in its details, is predictable in its general directions when selective pressures are sufficiently strong. The history of eye evolution also illustrates the difference between evolving a new structure and modifying existing ones. Despite the structural difference between vertebrate and arthropod eyes, both use the same basic photosensitive pigment, opsin, to detect light. That molecular similarity, despite macroscopic morphological difference, is evidence that the eyes of all animals descend from the same original evolutionary innovation, probably a simple photosensitive cell from the Cambrian or earlier. Among all the Cambrian creatures, one has special importance for us humans. Not because it was the largest, or the most spectacular, or the most abundant, but because it was the ancestor of all vertebrates, Pichia. Pichia was a small animal, barely 5 centimeters in length, with a flattened body and a form recalling a primitive fish, though without the fins or jaws of modern fish. Its most important characteristic, the one connecting it to vertebrates, was the presence of a notochord-like structure, the tube of firm tissue that in vertebrates develops into the spinal column. The notochord gave Pichia body stiffness and allowed it to swim with an undulating motion, the same fundamental motion that fish use today for propulsion. The story of Pichia also has an interesting scientific dimension beyond its evolutionary importance. For decades, Pichia was considered the only known Cambrian chordate, based on Burgess Shale specimens. That position of being the only known representative of its group in the Cambrian record made Pichia extraordinarily important but also extremely vulnerable to reinterpretation. When Changjiang specimens revealed similar but different animals, and when more detailed analysis of Pichia fossils suggested it might not be exactly the first chordate but one among several, the image of Pichia as the sole ancestor of all vertebrates became more complex. Today we know Pichia was probably just one among several primitive Cambrian chordates, which makes the history of vertebrate origins less contingent on any single species but raises its own fascinating questions about the diversity of early chordate experiments. The Pichia and its fellow early chordates also illustrate the importance of developmental genetic innovations in enabling major evolutionary transitions. The evolution of the notochord, the hollow dorsal nerve cord, and the pharyngeal slits that define the chordate body plan each required specific genetic changes. Studies of the developmental genetics of modern Amphioxus have revealed that many of the genes controlling notochord development in modern vertebrates are present in ancestral forms in Amphioxus, confirming that the chordate body plan was assembled from pre-existing genetic components in the Cambrian or before. That modular assembly of new body plans from existing genetic components is a recurring theme in evolutionary innovation. The connection between Pichia and all subsequent vertebrate evolution is made particularly vivid when considering the scale of what descended from that 5-centimeter animal. All 450 species of sharks, all 33,000 species of bony fish, the 7,000 species of amphibians, the 10,000 species of reptiles, the 10,000 species of birds, and the 5,500 species of mammals alive today, all of them are the evolutionary descendants of Cambrian animals not very different from Pichia. That genealogy, from a 5-centimeter soft-bodied Cambrian chordate to the full diversity of vertebrate life on Earth today, is one of the most extraordinary evolutionary stories that can be told. The relationship between Pichia and all subsequent vertebrate evolution is also a reminder of how the most consequential events can be the least conspicuous at the time they occur. In a Cambrian ocean filled with spectacular large predators and strange armored creatures, the small soft-bodied Pichia was probably among the least impressive organisms present. No paleontologist from the future, arriving in the Cambrian Ocean with no knowledge of what would follow, would have identified that small undulating swimmer as the ancestor of all vertebrates. Evolution's most important transformations often begin in the margins, with the inconspicuous and the easily overlooked. The modern Amphioxus, the lancelet, remains the best living analog of what the Cambrian chordate ancestors of vertebrates looked like. Its biology and genetics have been intensively studied because they provide clues about what that ancestor was like. Amphioxus comparative genomic studies have confirmed that vertebrates evolved from an ancestor similar to Pichia, with massive genome duplications enabling the additional complexity of vertebrates. When Stephen Jay Gould wrote about Pichia in his book Wonderful Life, he posed a question that became one of the most famous thought experiments in evolutionary biology. If we could rewind the tape of evolution and play it again, would vertebrates reappear? Would humans reappear? Gould argued that the contingency of evolution, the dependence of outcome on historical accidents and chance, made it improbable that the same result would repeat. Picaia, that 5-centimeter wormfish swimming in the Cambrian, could perfectly well have been eliminated in one of the Paleozoic extinctions, and if it had been eliminated, vertebrates would not have existed, nor mammals, nor primates, nor humans. The debate between Gould and Conway Morris over the significance of Burgess Shale fauna was not only a disagreement about data, but a disagreement about how to interpret that data within different theoretical frameworks. Conway Morris, who worked with Gould on the reinterpretation of Burgess Shale fossils but reached very different conclusions, has argued that convergent evolution is so common and so predictable that the general results of evolution were somehow written in the physical and chemical laws. Although not necessarily humans, he argues, something with intelligence comparable to the human would inevitably have emerged if evolution had sufficient time and basic conditions were adequate. The genomic studies that revealed the history of vertebrate genome duplications are one of the most important contributions of molecular biology to the knowledge of Cambrian evolution. Without those studies, the story of vertebrate origins would have remained obscure, because the fossil record of the first vertebrates is scarce and fragmentary. Comparative genomics has allowed reconstructing aspects of that history that fossils cannot reveal. Vertebrates have genomes approximately 4 times larger than those of their closest non-vertebrate relatives, the Amphioxus, reflecting 2 successive rounds of complete genome duplication that occurred in the first Cambrian vertebrates or shortly after. To study the Cambrian, paleontologists need exceptional sites, places where preservation conditions were so favorable that even the soft tissues of organisms were recorded in the rocks. The most famous of all those exceptional sites is the Burgess Shale in the Rocky Mountains of British Columbia, Canada. The history of scientific interpretation of the Cambrian is also a story about how scientific knowledge changes when new evidence becomes available. Walcott's interpretations in the 20th century, which classified most Burgess Shale fauna as primitive ancestors of known modern groups, were conservative but not irrational given the knowledge available in his era. The reinterpretations of the 70s and 80s, which revealed the strangeness and diversity of Cambrian fauna, were possible because preparation and observation techniques had improved and because the paleontological community had developed more sophisticated conceptual frameworks for interpreting fossils. The Burgess Shale also represents one of the most important arguments for the conservation of geological heritage. The fossils contained in it cannot be replaced if destroyed. A trilobite fossil from a rock quarried for construction represents the permanent loss of scientific information that can never be recovered. The recognition of that irreplaceable nature of exceptional fossil sites has driven the development of international frameworks for the protection of paleontological heritage, of which the World Heritage designation of both the Burgess Shale and Chengjiang are among the most important examples. The Burgess Shale represents something philosophically profound as well as scientifically important. A reminder that the world visible to us in any given moment is only a fraction of the full reality that has existed. The organisms of the Burgess Shale were invisible to human knowledge for 508 million years. They were there, preserved in rock, waiting to be found and understood. How many other realities, past and present, are we currently invisible to? How much of existence is outside the frame of our current perception and knowledge? The Burgess Shale is one answer to the question of what exists outside that frame. The cultural impact of the Burgess Shale discovery extends beyond science into a broader shift in how educated people think about the history of life. Before the Burgess Shale became widely known through popular science writing, the standard narrative of evolutionary history was one of progress. From simple to complex, from primitive to advanced, from the past to us as the culmination. The Burgess Shale, with its suggestion that the Cambrian contained more body plan diversity than exists today and that subsequent evolution was as much elimination as progress, challenged that narrative in a fundamental way. The history of life is not a march toward us. It is an exploration of a vast space of biological possibilities, of which the present is just one sample. The preparation techniques for Burgess Shale fossils have improved enormously since Walcott's time. Preparation under microscope with ultrafine air tools can reveal details previously invisible. Selective chemical staining can make visible differences in the composition of different fossil parts not visually distinguishable. And photography with different illumination types, including ultraviolet illumination that makes some preserved organic materials fluorescent, has revealed details in specimens that had been studied for decades without detecting them. That history of revision illustrates a general principle about scientific knowledge. Interpretations are always limited by available techniques and existing conceptual frameworks. What seems a definitive interpretation at one moment can be deeply revised when new evidence or new techniques allow seeing data differently. In paleontology, where data are inevitably incomplete and organisms often radically different from anything known, that revisability is especially important. The Burgess Shale was discovered in 1909 by paleontologist Charles Doolittle Walcott, who at that time was secretary of the Smithsonian Institution, the world's largest museum institution. Walcott was exploring the Rocky Mountains when he found a split stone showing the fossil of a perfectly preserved marine arthropod with the body's soft parts visible in the rock. That discovery led him to explore the rock wall from which the stone had fallen, and there he found one of the world's richest fossil sites. Later research in the Burgess Shale has continued producing new discoveries decades after Walcott's original work and the reinterpretation of the 1970s and 80s. New excavation areas have produced new species, and the application of modern techniques to old specimens has revealed anatomical details not visible with earlier techniques. The Burgess Shale remains one of the world's most actively investigated sites and seems likely to continue producing surprises for many decades. The techniques used to study Cambrian fossils have improved enormously in recent decades. High-resolution computed tomography allows visualizing the three-dimensional internal structure of fossils without damaging them. Scanning electron microscopy reveals details of fossil surfaces at micrometric scale. Spectroscopic analysis can identify the chemical composition of fossilized tissues, sometimes revealing the presence of pigments or specific organic compounds, providing information about organism physiology. The preservation of Burgess Shale fossils was a consequence of exceptional depositional conditions. Organisms living on a shallow carbonate platform were periodically swept by turbidity currents into deeper anoxic waters, where the absence of oxygen prevented decomposition of soft tissues. Those exceptional preservation conditions are what makes the Burgess Shale so valuable. It preserves not only the shells and skeletons that normally fossilize, but also intestines, eyes, gills, muscles, and other soft tissues that normally disappear without trace. The impact of the Burgess Shale on paleontology goes far beyond the specific organisms it contains. It is also a model for how exceptional preservation can radically transform our understanding of life's history. Before the Burgess Shale, most Cambrian knowledge came from mineralized skeletons and shells, which represent a small fraction of all organisms living in that period. The Burgess Shale revealed that most Cambrian diversity, in terms of species number and ecological diversity, was in soft-bodied organisms that normally do not preserve. The Burgess Shale is not the only extraordinary window into the Cambrian. In recent decades, a Chinese site has transformed our understanding of the period in a way no other recent paleontological discovery has matched: the Changjiang site in Yunnan Province, southwestern China. The Changjiang fauna also includes some of the most important evidence for the evolution of nervous systems in the Cambrian. Some Changjiang fossils preserve traces of what could be brains or ganglia in the organisms containing them, providing the oldest world evidence of centralized nervous system development in animals. Those traces, though difficult to interpret with certainty, are consistent with the idea that complex and centralized nervous systems, one of the most important characteristics of modern vertebrates, began their evolution in the early Cambrian. The Changjiang biota also includes organisms that appear to represent intermediate forms between recognized phyla, providing evidence for the evolutionary transitions between major animal groups that are often unclear from the fossil record of later periods. Those intermediate forms are possible to recognize in Changjiang because the fauna preserves organisms at a stage of evolution when the differentiation between phyla was less complete, when organisms that would eventually become clearly distinguishable as members of different phyla still retained characters of their common ancestors. Studying those transitional forms is one of the most direct ways to understand how the major animal lineages diverged from each other in the Cambrian. The Changjiang biota also reminds us that our understanding of the Cambrian, while remarkable in its depth, is almost certainly still incomplete in important ways. The organisms we know from Changjiang and the Burgess Shale lived in particular environments under particular conditions. The organisms that lived in different environments, the open ocean swimmers, the deep sea inhabitants, the organisms of polar seas and shallow reefs in different parts of the world, are mostly unknown to us. The Cambrian we know is a sample, not the complete picture. The Changjiang site also demonstrates something important about the relationship between paleontology and geopolitics. The intensive Chinese investment in paleontological research in the decades since Changjiang's discovery has transformed China into one of the world's leading centers of paleontological research. That transformation reflects not only the scientific importance of Changjiang, but also China's recognition that understanding the deep history of life on its territory is an important component of scientific and cultural heritage. The rich record of Cambrian organisms from China has also highlighted the global nature of the Cambrian Explosion. The same types of organisms, the same ecological roles, the same predator-prey dynamics appear in Cambrian rocks from China, Canada, Morocco, Greenland, Australia, and many other regions. That global simultaneity of the explosion across all the world's oceans in a geologically brief interval confirms that the Cambrian was driven by factors affecting the entire planet, not local conditions in particular regions. The collaboration between Chinese and international teams in the study of Changjiang has been one of the most successful cases of international scientific cooperation in recent paleontology. That collaboration has produced a stream of discoveries that would not have been possible through either national tradition alone, demonstrating the value of international scientific exchange in advancing our understanding of fundamental questions about the history of life. Changjiang was discovered in 1984 by Chinese paleontologist Ho Xinguang, who found the first fossil while looking for brachiopods in the Yellow Hills of the region. What he found was a perfectly preserved fossil of an arthropod with soft parts visible, the Nereoia, known only from the Burgess Shale. That finding indicated Changjiang contained fauna comparable to the Burgess Shale but potentially older. Later studies confirmed the Changjiang fauna is approximately 5 million years older than that of the Burgess Shale, making it one of the oldest known well-preserved records of early Cambrian fauna. Among the most important Changjiang organisms are the first known vertebrates. Haikuichthyes and Milocunmingia are two small animals approximately 3 centimeters in length interpreted as the most primitive known vertebrates, with a differentiated head, possible branchial arches, and a well-defined notochord. The Changjiang site has also had a significant impact on the relationship between China and international paleontology. Before Changjiang's discovery, the most important Cambrian sites were in North America and Europe, and Cambrian paleontology was dominated by researchers from those regions. Changjiang transformed that scientific geography, placing China at the center of the most important Cambrian paleontology debates and establishing Chinese paleontologists as leaders in that field. The Changjiang site also produced evidence of complex behaviors in Cambrian organisms that could not be inferred from skeletons alone. Some specimens preserve intestinal contents, allowing knowledge of the animal's exact diet. Others preserve predator bite marks, allowing inference of specific predator-prey relationships in that ecosystem. And some preserve evidence of gregarious behavior, of organisms moving in groups, which are the oldest hints of social behavior in the fossil record. The importance of Changjiang for world paleontology was recognized by UNESCO when it designated the site as World Heritage in 2012, making it one of the few paleontological sites in the world with that recognition. The systematic excavation of Changjiang has produced thousands of specimens of more than 200 species, including many known from no other site. The deepest question about the Cambrian explosion is not what animals lived in that period. It is why it happened when it happened. Complex life had been existing, in the form of algae and microbes, for more than 2 billion years. The first multicellular animals appear in the fossil record at least 100 million years before the Cambrian. Why did the explosive diversification of animal body plans occur precisely in the early Cambrian and not before? The genetic toolkit for building animal bodies, though modified and elaborated over 500 million years, still carries clear traces of its Cambrian origins. The Hox genes that control the anterior-posterior axis of animal development in all bilaterian animals are directly homologous across all groups, from insects to vertebrates. A mouse Hox gene transplanted into a fly can control the correct development of fly body segments, because the genetic instructions for that fundamental body axis were written once, in the Cambrian or before, and have been conserved through all subsequent evolution. The Cambrian explosion also demonstrates the importance of ecological opportunity in driving evolutionary diversification. The Cambrian Ocean was in many ways a blank slate ecologically, with many potential niches for complex animals that had never been occupied. That abundance of ecological opportunity, combined with the genetic plasticity of early animal lineages, allowed diversification to proceed at rates that have never been matched since. Understanding the relationship between ecological opportunity and evolutionary diversification rate is one of the central questions in macroevolution, and the Cambrian provides the most striking example of what happens when those conditions are fully met. The Cambrian explosion also illustrates the importance of threshold effects in complex systems. For hundreds of millions of years, life existed below some threshold of complexity, and the world looked essentially the same from 1 million years to the next. Then several factors converged: oxygen levels, ocean chemistry, ecological opportunity, and perhaps developmental genetic changes, and the system crossed a threshold above which diversification could accelerate in ways that maintained themselves. Understanding those threshold effects in the Cambrian may provide insight into other complex systems, from ecosystems to economies, where long periods of apparent stability are punctuated by rapid and transformative change. The Cambrian explosion also holds lessons about the relationship between diversity and resilience. The extraordinary diversity of body plans and ecological strategies produced in the Cambrian created an ecosystem that was, paradoxically, both fragile and robust. Fragile in that any individual lineage, including the ancestral vertebrates, was vulnerable to extinction from environmental perturbation. Robust in that the overall system, with its many redundant roles and multiple solutions to the same ecological problems, could absorb the loss of individual lineages without collapsing. That relationship between diversity and resilience is one of the central principles of modern ecology, and the Cambrian produced the first large-scale demonstration of it. That deep conservation of developmental genetics is both a testament to the fundamental importance of those genetic programs and a constraint on subsequent evolution. Because those gene networks are so deeply integrated into development, modifying them radically tends to be lethal. The result is that the basic body plans established in the Cambrian, though endlessly modified in detail, have not changed fundamentally in 500 million years. The limits of evolutionary creativity at the level of fundamental body architecture were set in the Cambrian. The climate models of the Cambrian, combining the position of continents, atmospheric composition, and ocean floor topography to simulate atmospheric and oceanic circulation, have provided predictions that can be compared with the geological record. When model predictions about sediment-type distribution, water temperatures inferred from isotopes, and fauna distribution coincide with geological evidence, that gives confidence that the model correctly captures Cambrian conditions. Hypotheses to explain the Cambrian explosion are numerous and largely complementary. There is no single generally accepted cause, but a set of factors that probably acted in combination to make the explosion possible and trigger it. One of the most influential hypotheses is the oxygen threshold. For most of the Precambrian, oxygen levels in the atmosphere and oceans were too low to support the active metabolisms of complex animals. The Cambrian explosion also raises questions about whether similar phenomena could have occurred or might occur on other planets with life. If on Earth life took more than 3 billion years to produce complex animals, and then experienced an explosive diversification when conditions were right, is there any reason to think that pattern would be different on another planet with life? Astrobiology considers the Cambrian as a potential model for what might occur on planets with biological histories similar to Earth's, though with completely different organisms. The connection between the geochemical changes of the late Ediacaran and early Cambrian, with their carbon isotope excursions and changes in marine strontium composition, and the biological explosion of the Cambrian is the subject of active investigation. The models attempting to establish causal relationships between those geochemical changes and the biological diversification must deal with the complexity of distinguishing cause from effect in a system where biological and geochemical changes were probably feeding back on each other. The appearance of organisms with calcite shells changed ocean chemistry. Changes in ocean chemistry favored the evolution of calcite shells. Separating those intertwined causalities is one of the most difficult challenges in Cambrian research. The oxygen threshold hypothesis proposes that in the Cambrian, oxygen levels first reached a threshold above which complex and active animals could function efficiently, triggering their diversification. The ecological trigger hypothesis proposes that the appearance of the first active predators was the event that triggered the explosion. The geochemical changes in the early Cambrian ocean are also relevant. The increase in dissolved calcium, facilitating skeleton mineralization, and changes in the isotopic composition of carbon and strontium record deep changes in ocean chemistry that could have created favorable conditions for diversification of animals with skeletons. The Cambrian was also the period of the first reefs in the history of life. Not coral reefs as we know them today, but reefs built by organisms called archaeocyaths, an extinct group that flourished during the early Cambrian and was the first successful experiment in life's history in building complex three-dimensional structures on the seafloor. The story of archaeocyath reefs is also relevant to modern conservation biology in ways that extend beyond historical interest. The pattern of reef ecosystem collapse at the end of the early Cambrian, driven by changes in ocean chemistry, provides one of the earliest examples of how sensitive reef ecosystems can be to environmental change. Modern reef ecologists studying the responses of coral reefs to ocean warming and acidification are essentially watching a replay of dynamics that first appeared in the Cambrian, though with different organisms and different proximate causes. The Cambrian fossil record thus provides perspective on the long-term vulnerability of reef ecosystems that cannot be obtained from historical observation alone. The Cambrian also introduced a new kind of ecological engineering that fundamentally changed the long-term trajectory of marine evolution, the production of biogenic calcium carbonate at large scale. Before the Cambrian, calcium carbonate was deposited primarily through abiotic precipitation from seawater. In the Cambrian, organisms began extracting calcium carbonate from the water to build their shells and skeletons, fundamentally altering the carbonate chemistry of the ocean and establishing the basis for the enormous carbonate rock record of the Phanerozoic. The limestone that forms much of the world's mountain ranges today was produced ultimately by organisms following in the biological tradition established by the first Cambrian shelled organisms. The first reefs of the Cambrian also established a pattern that has persisted throughout geological history. The tendency for reef ecosystems to collapse during periods of ocean chemistry perturbation and then to be rebuilt by new organisms with different body plans but similar ecological roles. The Cambrian archaeocyath reefs were replaced by Ordovician coral and sponge reefs, which were replaced by Devonian reefs of a different composition, which were replaced by Permian reefs, each time after an extinction event wiped out the previous reef builders. Modern coral reefs are the latest version of that recurring pattern, and understanding the Cambrian precedent helps put their current vulnerability in long-term context. The Archaeocyath reefs also illustrate an important principle about the relationship between ecological engineering and biodiversity. Complex three-dimensional structures created by ecosystem engineers harbor more species than simple flat substrates. The Archaeocyath reefs of the early Cambrian, by creating a topographically complex substrate in what had previously been flat sediment, immediately increased the number of available ecological niches and attracted a diverse community of organisms. That principle, that structural complexity created by ecosystem engineers promotes biodiversity, has been one of the most consistent patterns in ecological research. The Cambrian explosion of body plans also produced the first organisms capable of engineering their environment in complex ways. Archaeocyaths not only built physical structures, they also altered the chemical environment around them by extracting calcium carbonate from the water, changed the hydrodynamics of water flow through their porous structures, and created chemical gradients that influenced what organisms could live near them. That capacity to engineer the local environment was a genuinely new ecological capability in the Cambrian, adding another dimension to the already complex ecological interactions of that extraordinary period. Archaeocyaths were organisms with cup or vase-shaped bodies, with double walls perforated by pores through which water circulated. Their basic structure superficially resembled modern sponges, and for many years they were classified as primitive sponges. More recent analyses have separated them from sponges and placed them as their own group within animals, possibly related to sponges but constituting an independent lineage. The extinction of Archaeocyaths at the end of the early Cambrian is also an example of how reef-building organisms can be devastated by relatively rapid changes in ocean conditions. Changes in temperature, water chemistry, or oxygen levels that may seem modest in climatic terms can be lethal for organisms that depend on specific conditions to calcify their skeletons. Modern coral reefs, being damaged by the warming and acidification of today's oceans, are experiencing something analogous to what Archaeocyaths experienced at the end of the early Cambrian, though at very different speeds. The Archaeocyath reef builders also provide evidence of possible symbiosis in Cambrian animals. There are indications that some Archaeocyath reefs harbored communities of symbiotic algae, similar to the symbiosis between modern corals and zooxanthellae. If that interpretation is correct, photosymbiosis in reef-building animals has its roots in the Cambrian, 500 million years before the modern corals that are the best-known example of that symbiosis. The history of Archaeocyaths is also a story about innovation and extinction. These organisms were the first to solve the problem of building a rigid three-dimensional structure in a marine environment, a problem that life had left unsolved for more than 3 billion years. That the first experiment in reef building became extinct relatively soon does not diminish the achievement. Archaeocyaths established the principle that organisms could build habitat for other organisms, a principle that evolution has reused repeatedly ever since. The ecology of the Cambrian was fundamentally different from that of any modern ecosystem, not only in the players but in the structure of the relationships among them. It was an ecosystem under construction, a world where ecological roles were being invented at the same time as the organisms performing them, where natural selection was simultaneously exploring a space of ecological possibilities that had not previously existed. The introduction of bioturbation in the Cambrian had consequences that extended far beyond the simple mixing of sediment. By creating channels and burrows in the sediment, the first burrowers changed the redox chemistry of the seafloor, allowing oxygen to penetrate deeper into the sediment and altering the distribution of chemical species at the sediment-water interface. Those chemical changes created new habitats, new gradients of resource availability, and new opportunities for organisms that could exploit them. The modern seafloor, with its complex community of burrowers, filterers, and surface dwellers all partitioning the available resources, is the descendant of the first simple partitioning that began in the Cambrian. The relationship between the Cambrian and the origin of animal consciousness is one of the most philosophically interesting questions raised by that period. The first neurons appeared well before the Cambrian, but the organization of neurons into complex centralized brains capable of integrating information from multiple sensory systems and generating complex responses appears to have its first clear expression in the Cambrian. If consciousness in some form requires a nervous system of sufficient complexity and integration, the Cambrian may have been the period when the first animals experienced something like a subjective perspective on the world. That possibility, impossible to test from fossils alone but suggested by the complexity of the nervous systems that the Cambrian appears to have first produced, gives the Cambrian an additional dimension of significance. The Cambrian also produced the first organisms capable of active locomotion in 3 dimensions, organisms that could choose to swim up, down, left, or right rather than simply moving along a substrate. That 3-dimensional mobility was not only ecologically transformative, opening up the entire water column as inhabitable space, but cognitively demanding. Navigating 3-dimensional space requires more sophisticated spatial processing than navigating a 2-dimensional substrate, and the evolution of 3-dimensional mobility in the Cambrian likely placed new demands on the nervous systems of the organisms that achieved it. The Cambrian also produced some of the first evidence of parasitism in the animal fossil record. Some Cambrian arthropod fossils show unusual structures on their bodies that have been interpreted as the attachment points of parasitic organisms. If those interpretations are correct, the parasitic lifestyle, one of the most ecologically important and most evolutionarily productive relationships in the history of life, began in the Cambrian, as a direct consequence of the intimate coevolutionary relationships that the Cambrian arms race generated. Parasitism is in some ways the ultimate form of ecological intimacy, and the Cambrian, the period that invented ecological intimacy between complex animals, was the natural birthplace of parasitism as well. The origin of planktonic feeding, organisms swimming or floating in the water column and filtering food from it, was another Cambrian ecological innovation with profound long-term consequences. Before the Cambrian, the planktonic food web was simple, dominated by microbes and their direct consumers. In the Cambrian, the first animals capable of actively filtering plankton from the water column appeared, connecting the microbial loop of the plankton to the larger animal food web in a new way. That connection transformed the efficiency with which energy and materials were transferred from primary producers to larger animals throughout the ocean. Bioturbation of sediment, the process by which burrowing and moving organisms alter the physical and chemical structure of sediment, was one of the most important ecological innovations of the Cambrian. Before the Cambrian, seafloors were covered with microbial mats forming firm structured surfaces. The first Cambrian burrowing animals disturbed those mats and began mixing the sediment, which had profound consequences for seafloor chemistry and nutrient availability. The introduction of active predation in the Cambrian also changed the relationship between organisms and the substrate. Before predation, ediacaran organisms lived on the microbial mat surface, safe insofar as nothing actively hunted them. With the appearance of predators, the substrate became a refuge. Burrowing into sediment to escape predators swimming in free water was a defensive strategy that many Cambrian organisms adopted. That transition from life on sediment to life in sediment was another of the fundamental ecological changes of the Cambrian, with consequences extending to today in the structure of marine benthic ecosystems. The carbon isotopes in Cambrian fossils also record the diet of the organisms that produce them, through the isotopic discrimination that occurs at each step in the food chain. By measuring the carbon and nitrogen isotopic ratios in fossilized tissues of Cambrian organisms, geochemists can in some cases reconstruct the trophic position of those organisms in the food chain, confirming or refuting interpretations based on the morphology of feeding organs. The apex of the Cambrian food chain was occupied by the active predators, with Anomalocaris and its relatives in the role of super-predators. The existence of active predators created selective pressures that had not existed before the Cambrian and drove the evolution of defenses in prey. That arms race between predators and prey was probably one of the most important drivers of Cambrian diversification, producing in millions of years a variety of defensive and offensive strategies that filled the ocean with new forms. The Cambrian was not a period of continuous, uninterrupted diversification. Throughout its 60 million years, the period was traversed by at least 3 significant extinction events that temporarily interrupted diversification and eliminated some of the groups that had prospered in earlier phases. The transition from the Cambrian to the Ordovician, occurring approximately 485 million years ago, was followed by one of the greatest evolutionary radiations in life's history, the Great Ordovician Biodiversification Event. In the Ordovician, marine diversity approximately tripled, with the appearance and diversification of rugose corals, bryozoans, graptolites, articulated echinoderms, and many other groups. That radiation was possible in part because the Cambrian had established the fundamental body plans and basic ecological innovations on which to build. The Cambrian-Ordovician transition also marks an important change in the dominance of different groups of organisms in marine ecosystems. The Cambrian was dominated by organisms with Cambrian-type ecological roles: mobile predators and prey, filter feeders, and burrowers. The Ordovician saw the rise of organisms with what paleontologists call Paleozoic-type ecological roles: attached suspension feeders like crinoids, bryozoans, and brachiopods that dominated the seafloor in a very different way from the mobile Cambrian fauna. That ecological transition, from a mobile Cambrian ecosystem to a more stationary Paleozoic ecosystem, fundamentally changed the structure of marine communities for the next 200 million years. The survival of the vertebrate lineage through the late Cambrian extinctions was itself not guaranteed. The chordate clade, from which all vertebrates descend, was represented by relatively few species in the Cambrian, none of them large or ecologically dominant. Any of the extinction events of the Cambrian could in principle have eliminated those few species and ended the vertebrate lineage before it began. That the lineage survived, diversified into the jawless fish of the Ordovician, and eventually gave rise to the full diversity of vertebrates is in part a matter of historical contingency, a reminder that the world as we know it was not inevitable. The late Cambrian extinctions are also important for understanding the relationship between extinction and evolutionary opportunity. The groups that went extinct in the late Cambrian vacated ecological space that was subsequently colonized by new organisms in the Ordovician. The bryozoans, the crinoids, the graptolites, and the rugose corals that came to dominate the Ordovician seafloor all exploited ecological space that had been made available by the late Cambrian extinctions. Without those extinctions, the Ordovician radiation might not have been possible, or might have taken a very different form. The relationship between late Cambrian extinction and Ordovician radiation is an example of the general pattern of extinctions followed by diversifications characterizing life's history. Extinctions eliminate established groups and create available ecological space. That available ecological space can then be colonized by new groups that diversify rapidly to fill it. The pattern of explosive diversification that followed the late Cambrian extinction is not qualitatively different from that following the 5 great Phanerozoic extinctions. It is simply an earlier version of the same process. The fossil record of the late Cambrian vertebrates, though sparse, shows a gradual increase in diversity through the period. By the end of the Cambrian, several distinct lineages of jawless fish were already differentiated, each exploring slightly different ecological niches. That diversity at the end of the Cambrian set the stage for the Ordovician radiation of jawless fish that would eventually lead to the first jawed vertebrates of the Silurian and Devonian. The most important of those events was the Bathomian-Cambrian extinction, approximately 510 million years ago, which eliminated most archaeocyoths and produced an important reorganization of early Cambrian ecosystems. That extinction is recorded in the geological record as a disruption of the carbon cycle, evidenced by a negative excursion in the carbon isotopes of marine carbonates indicating a massive release of organic carbon to the system. The absence of jawed vertebrates in the Cambrian is also informative about the pace of evolution after the explosion. Jawless vertebrates of the Ordovician and Silurian took millions of years to evolve jaws, one of the most important innovations in vertebrate history. That latency time between the establishment of the vertebrate body plan in the Cambrian and the appearance of some of its most important innovations in later periods suggests the Cambrian explosion was the beginning of a process that took much longer to complete. The late Cambrian extinction also illustrates in terms of what survived and what did not. Groups that survived best were generally those most broadly distributed geographically, least specialized in terms of habitat and diet, and most capable of recovery after disturbances. Those are exactly the same factors predicting survival in more recent mass extinctions, suggesting the basic principles of extinction vulnerability were established from the first Cambrian extinction events and have remained constant throughout all subsequent history of complex life. The end of the Cambrian was also the moment when vertebrates began their long road toward dominance of terrestrial ecosystems. The small chordates of the early Cambrian, like Pikaia and Haecuichthys, evolved through the late Cambrian and Ordovician into the first jawless fish forms. Those first jawless fish would be the ancestors of all modern jawed vertebrates, including sharks, bony fish, amphibians, reptiles, birds, and mammals. One of the most powerful ideas emerging from Cambrian study is that of body plans. The concept of a body plan, or BA-plan in comparative biology terminology, refers to the fundamental architecture characterizing a group of animals. The number and arrangement of their body parts, the symmetry of their body, the presence or absence of certain structures like a differentiated head, segmentation, or an internal or external skeleton. The idea that the Cambrian was the only period in which new fundamental body plans were invented has a deep philosophical implication. It suggests that evolution has creative limits that are set early in the history of a group and become difficult to overcome afterward. Once a body plan is established, with its developmental genes, its morphogenetic pathways, its tissue interactions, radically modifying it tends to produce non-viable organisms. The plasticity that existed in the first complex animals, when body plans were not yet completely integrated, no longer exists in modern animals with their 500 million years of evolutionary refinement. The study of the Cambrian has also transformed our understanding of what the beginning of complex animal life was actually like. Before the modern era of Cambrian paleontology, beginning with the reinterpretation of the Burgess Shale in the 1970s, the Cambrian was imagined as a period of progressive improvement, with primitive simple ancestors gradually giving way to more complex modern forms. The picture that has emerged from modern research is very different, a period of extraordinary experimentation, with many more body plans and ecological strategies being tried and discarded than survived, more similar to an evolutionary brainstorming session than to a progressive march toward the present. The Cambrian also demonstrates that the most important events in life's history are often invisible at the time they occur. The organisms of the Cambrian had no awareness of participating in the most significant biological transition in the history of complex life. They were simply living, competing, surviving, and reproducing in the ocean they knew. The significance of what they were doing, the establishment of the body plans that all subsequent animal life would elaborate, was entirely invisible from inside the Cambrian. That invisibility of the most important events from the perspective of those living through them is perhaps a universal feature of transformative moments. The Cambrian body plan explosion also illustrates the concept of evolutionary irreversibility in a striking way. Once a body plan is established with its associated genetic programs and developmental cascades, it cannot be undone. A vertebrate cannot evolve back into a non-vertebrate. An arthropod cannot shed its exoskeleton plan and develop an endoskeleton. The choices made in the Cambrian, in some sense, were permanent choices. The body plans invented then became the permanent templates for all subsequent vertebrate, arthropod, molluscan, and echinoderm evolution. The Cambrian established boundaries that have not been crossed in 500 million years. The Cambrian also poses questions about what life would look like if the same conditions occurred on a different planet. The same chemical and physical principles that governed Cambrian evolution would operate. The same ecological dynamics of predation driving defense, of filter feeding exploiting primary production, of burrowing modifying the substrate, would probably appear. But the specific organisms, their body plans, their behaviors, would be entirely different. Life on another planet with a Cambrian-like event might produce ecosystems with the same functional structure as the Cambrian, but with organisms recognizable as neither vertebrates, arthropods, nor any other group we know. The fascination the Cambrian exercises over scientists and the general public also has a philosophical dimension worth exploring. The Cambrian raises questions about contingency and necessity in evolution, about whether the current world was the only possible outcome or simply one among many. If the Cambrian explosion had produced a different set of body plans, if chordates had not survived the Cambrian extinctions, if some other group had dominated vertebrate evolution, the current world would be radically different. That contingency of the Cambrian, that dependence of the outcome on historical accidents, is both a source of wonder and of humility. Today we recognize approximately 35 animal phyla, the great groups representing fundamentally different body plans. Mollusks, with their soft body and mantle. Arthropods, with their exoskeleton and articulated appendages. Echinoderms, with their pentaradial symmetry. Chordates, with their notochord and dorsal nerve cord. Each of those groups has its own body plan distinguishing it from all others. The fossil record of the Cambrian shows that almost all those body plans appeared in a relatively brief interval of time, in the first 20 or 30 million years of the period. That is what makes the Cambrian explosion so extraordinary. Not only that many new species appeared, but that new body plans appeared, new fundamental architectures of animal bodies, in a way that has not occurred again in the subsequent 500 million years. The Cambrian also raises questions about contingency and necessity in evolution that go beyond biology and touch philosophical questions about the nature of causality and predictability. If evolution were completely deterministic, if the same genes and the same environmental conditions always produced the same results, then the Cambrian and everything that followed was inevitable from the Big Bang. If evolution were completely random, there would be no predictable pattern and the world could have taken any direction. Reality lies somewhere between those extremes. There are predictable patterns driven by natural selection, but there is also historical contingency, dependence on accidents and initial conditions known only in retrospect. The literature on Cambrian body plans also raises questions about whether the space of possible animal body plans is truly finite. Are there only 35 possible fundamental architectures? Or are there others that could exist in principle but are inaccessible to evolution from the current body plans without passing through non-viable intermediate stages? The Cambrian suggests that most of the accessible stable points in that space were found and occupied in the first 20 million years of animal diversification. The study of the Cambrian depends fundamentally on sites of exceptional preservation, the so-called Lagerstätten, places where geological conditions allowed the preservation of soft tissues that normally do not fossilize. Without those sites, our understanding of the Cambrian would be completely different and far more incomplete. The fossil record of the Cambrian is also being enriched by discoveries in sedimentary environments not traditionally considered favorable for exceptional preservation. Recent work has shown that some siliceous sediments, phosphorite deposits, and even certain types of limestone can preserve soft tissue detail under specific geochemical conditions that were not previously recognized as favorable. Those discoveries suggest the potential for exceptional preservation is more widespread than previously thought, and that with appropriate search strategies, new Cambrian Lagerstätten may yet be found in many parts of the world. The Cambrian revolution in predator-prey dynamics also had long-term consequences for the evolution of animal cognition and behavior. The demands of predation, including locating prey, planning and executing attacks, and avoiding capture by other predators, placed premiums on faster information processing, better sensory integration, and more flexible behavioral responses. Those pressures began in the Cambrian and have been maintained in animal lineages ever since. The cognitive sophistication of modern animals, from the problem-solving of crows to the social intelligence of primates, is built on a foundation of neural adaptations whose selective origins trace back to the Cambrian arms race. The Cambrian Revolution also changed the physical structure of the seafloor in ways that had permanent geological consequences. The shells and skeletons of Cambrian organisms, accumulated on the seafloor over millions of years, were compacted and cemented into the carbonate rock that forms much of the geological record from the Cambrian onward. Before the Cambrian, carbonate rocks were relatively rare and formed mainly by abiotic chemical precipitation. After the Cambrian, biological carbonate production dominated the sedimentary record. The limestone of the Alps, the chalk of the English cliffs, the coral reefs of the Indo-Pacific, all are the descendants of the biological carbonate production that began in the Cambrian. The Cambrian explosion also raises fundamental questions about the pace of evolution. The standard model of evolution, with gradual change accumulating slowly over long periods, seems inadequate to explain the rapid appearance of so many new body plans in the early Cambrian. The alternative of punctuated equilibrium, with long periods of stasis interrupted by rapid bursts of change, seems more consistent with the Cambrian pattern. Modern evolutionary biology increasingly recognizes that evolutionary rates can vary enormously, from negligibly slow to surprisingly fast, depending on the intensity of selective pressures and the genetic variability available in populations. The paleobiology of Cambrian organisms is also being reconstructed through the study of ichnofossils, the trace fossils left by organism behavior, in ways that complement the information from body fossils. The complexity of Cambrian trace fossil assemblages increased dramatically through the period, from simple shallow horizontal traces in the early Cambrian to complex three-dimensional burrow systems in the late Cambrian. That increase in trace complexity is a proxy for increasing behavioral sophistication among Cambrian organisms, suggesting that the behavioral evolution of Cambrian animals was as remarkable as their morphological evolution. The documentation of Cambrian biodiversity has also been advanced by the application of statistical methods originally developed for ecology to the paleontological record. Those methods allow estimating the total diversity of Cambrian faunas from the known sample, correcting for the incompleteness of the fossil record. The results suggest that Cambrian diversity was substantially higher than the number of described species would indicate, and that many Cambrian organisms remain to be discovered and described. The digitization of Cambrian paleontological collections has also been an important advance in recent decades. Collections of the Smithsonian, the Royal Ontario Museum, the Natural History Museum of China, and other institutions have been partially digitized, creating databases that allow researchers to compare specimens from different collections without physically traveling to each institution. That digital accessibility of collections has accelerated research and democratized access to paleontological data, allowing researchers from countries with fewer resources to participate in Cambrian research in ways previously impossible. The geochemistry of rocks containing Cambrian fossils also provides crucial information about environmental conditions in which those organisms lived. Carbon isotopes record changes in ecosystem productivity and the carbon cycle. Sulfur isotopes record changes in ocean oxygen levels. Strontium isotopes record changes in continental erosion rates and seawater composition. Cambrian paleontology has also been transformed by the digital revolution in access to scientific information. Open access publications, online fossil databases, and online discussion forums have accelerated information exchange between researchers from different countries and institutions. A paleontologist in China finding a new organism can compare it with specimens from all world sites within hours, something requiring months or years of travel and mail correspondence in previous decades. The paleontology of the Cambrian in regions of the world not yet exhaustively explored promises new discoveries that could add additional dimensions to our understanding of the period. Mountain zones of Central Asia, the polar regions of the Arctic and Antarctica, and ocean floors where Cambrian rocks outcrop are territories paleontologists are beginning to explore more systematically. Each newly discovered site has the potential to reveal organisms, behaviors, and environmental conditions not known from existing sites, adding pieces to the Cambrian puzzle that after 150 years of intensive research remains fascinatingly incomplete. There is a connection between the Cambrian and human biology that goes deeper than any known evolutionary genealogy. We are vertebrates. We are chordates. And chordates are one of the body plans that appeared in the Cambrian Explosion. Every structure of our body, from the spinal column to the brain, from the eyes to the immune system, has its ultimate origin in that Cambrian radiation that produced the first complex animals. The applications of Cambrian research extend into medicine and material science in ways that were not anticipated when paleontologists first began studying these ancient organisms. The optical properties of trilobite calcite lenses, which are achromatic doublets correcting for spherical aberration, were independently discovered by humans only in the 17th century. The understanding of how trilobites solved those optical problems in the Cambrian has informed the design of specialized optical systems for scientific instruments. The connection between the Cambrian and modern human experience extends even to some of our most fundamental biological rhythms. The circadian clock, the internal 24-hour cycle controlling sleep and wakefulness, metabolism, and many other biological processes, is present in all animals and appears to be a very ancient adaptation. The selective pressures that maintained that clock in Cambrian animals were the same ones operating in all animals since, the advantage of synchronizing biological processes with the daily light-dark cycle. When we sleep tonight, we are responding to a rhythm that was already operating in the first complex animals of the Cambrian 540 million years ago. The connection between the Cambrian and the neuroscience of sleep is more than a metaphor. The genes controlling the circadian rhythm in modern animals include many that are also involved in the development and function of the nervous system. The same fundamental neural circuits that generate sleep and wakefulness in modern animals appear to be deeply conserved from the first animals with centralized nervous systems, whose origins trace back to the Cambrian. When the neurons that generate the sleep state activate tonight, they are using biochemical processes whose fundamental logic was established in the Cambrian Ocean 500 million years ago. The Cambrian also provides a context for understanding our own moment in geological history. We live in the Anthropocene, a proposed geological epoch defined by human impact on the planet. That impact includes a rate of species extinction estimated to be 100 to 1,000 times the background rate that prevailed before human civilization. In the context of the Cambrian Explosion, where new species appeared faster than they went extinct for millions of years, the current situation is its mirror image. Species going extinct faster than new ones can evolve. The Cambrian tells us what the world can produce when conditions are right for diversification. The present moment tells us how quickly that diversity can be lost. The Cambrian also produced something that is easy to overlook, the first animals capable of suffering. Pain, the subjective experience of harm, requires a nervous system complex enough to generate that experience. The first complex nervous systems of the Cambrian, while almost certainly simpler than the nervous systems of modern vertebrates, may have been complex enough to generate some form of nociceptive experience. The arms race between predators and prey that drove so much Cambrian evolution was not merely a mechanical competition of shells and claws. It may have been the first time in the history of life that there was something it was like to be hunted, something it was like to be caught. The study of biomineralization in Cambrian organisms has also provided insights relevant to the development of medical implants and tissue engineering scaffolds. Understanding how the first shelled Cambrian organisms controlled the deposition of calcite and other minerals in specific crystal orientations and geometries has informed efforts to create synthetic biomaterials with similar controlled properties. The Cambrian was the first time in Earth's history that organisms solved the problem of controlled biomineralization at scale, and that 500-million-year-old solution contains lessons still being learned today. The Cambrian also raises questions about the relationship between biological innovation and environmental change that are directly relevant to current concerns about biodiversity. The Cambrian explosion occurred at a time of significant environmental change, including changes in ocean chemistry, oxygen levels, and continental configuration. Whether those environmental changes were necessary preconditions for the explosion or merely coincidental with it is still debated. But the association between major environmental change and major biological innovation is a recurring theme in evolutionary history that informs our thinking about how biological communities respond to rapid environmental perturbations. The human brain, the organ allowing us to explore the Cambrian, think about evolution, and listen to this story tonight, is the elaboration of 500 million years of a nervous system whose roots are in the first Cambrian chordates. The central nervous system of vertebrates, with its brain at one end of the body surrounded by a protective skull and its spinal cord protected by the vertebral column, evolved from the dorsal nerve cord of the first Cambrian chordates. Every neuron currently processing the words of this documentary is the heir of the neurons of the first animals with a nervous system in the Cambrian. The Cambrian also raises questions about the possibility of life in other oceans of the solar system. The moons Europa of Jupiter and Enceladus of Saturn have liquid oceans under their frozen surfaces, kept liquid by geothermal heat produced by the tidal forces of the giant planets. If those oceans have hydrothermal vents and a nutrient supply, they could harbor microbial life forms. And if that microbial life has had sufficient time, could it have experienced its own equivalent of a Cambrian explosion? The connection between Cambrian and our existence as a species also extends to the materials surrounding us. The calcium carbonate forming the marble and limestone of buildings and monuments we build is largely the accumulated result of 500 million years of marine organisms with calcite skeletons, starting with Cambrian archaeocyaths. The oil and natural gas we extract from the subsoil are partly the result of the organic matter produced by Cambrian and later marine ecosystems. Even the oxygen we breathe is the result of 540 million years of photosynthesis, beginning with Cambrian phytoplankton. We are, in the most literal and most biological sense possible, the Cambrian looking at itself. The organisms of that 540 million-year-old ocean could not imagine what evolution would do with the body plans they were inventing. But that chain of transformations, that uninterrupted process of modification and elaboration beginning with Pikaia and the first trilobites and continuing today in every living organism on the planet, is the longest and most extraordinary story that exists. The Cambrian was not only the ocean of the Burgess Shale or Changjiang. It was a global period, with ecosystems at all latitudes and in all marine environments of the planet, each with its own variation on the central theme of the Cambrian explosion. The Cambrian faunas of South America, preserved in rocks of Venezuela, Bolivia, and Argentina, show connections with both North American and Gondwanan faunas that help reconstruct the paleogeographic history of those continents during the Cambrian. The trilobites of those South American Cambrian faunas include both endemic species found nowhere else and cosmopolitan species also known from other continents, reflecting both the isolation that produced endemic lineages and the connectivity that allowed cosmopolitan species to spread widely. The study of Cambrian fauna from Australia, particularly from the Emu Bay Shale in South Australia, has provided important insights into the Cambrian fauna of Gondwana. The Emu Bay Shale is approximately 514 million years old and preserves a fauna with excellent soft tissue preservation, similar in quality to the Burgess Shale but from a different part of the Cambrian world. The comparison between the Emu Bay Shale fauna and those of the Burgess Shale and Changjiang reveals both similarities, reflecting the global nature of the Cambrian explosion, and differences, reflecting the geographic differentiation that was already beginning among Cambrian animal communities. The paleogeographic story of the Cambrian is also a story about the origin of biogeographic patterns that persist, in modified form, to the present day. The faunal provinces established in the Cambrian, the different communities of organisms that developed in geographically isolated parts of the Cambrian Ocean, were the ancestors of the biogeographic regions we recognize in modern marine biology. The distinction between Atlantic and Pacific fauna, between tropical and temperate marine communities, between shallow water and deep sea assemblages, all have their ultimate origins in the differentiation of geographically separated Cambrian communities. The Cambrian also invites reflection on what it means to be at the beginning of something. The Cambrian animals were, literally, at the beginning of the history of complex animal life. They had no precedent, no model for what a complex animal could be or do. They were the experiment that produced the first answers to questions about how to build eyes, how to build shells, how to hunt, how to hide. Everything they tried was tried for the first time. That sense of origination, of being at the absolute beginning of a long story, is one of the most powerful things the Cambrian conveys. The polar Cambrian faunus, known from sites in Greenland, Spitsbergen, and Antarctica, are generally less diverse than the tropical ones but show their own interesting characteristics. The organisms living in those high-latitude Cambrian seas adapted to conditions of seasonal light availability and lower temperatures not experienced by their tropical counterparts. Studying those adaptations provides information about the physiological tolerances of Cambrian organisms and the range of environments they could inhabit. The ongoing exploration of the Cambrian in Central Asia, particularly in Kazakhstan and Uzbekistan, where Cambrian strata are extensive and largely unexplored by modern paleontological methods, promises significant new discoveries in coming decades. Those regions lay at paleolatitudes different from both the tropical Burgess Shale environment and the Gondwanan-Changjiang environment, and might preserve fauna with their own distinctive characteristics reflecting conditions in the intermediate latitude Cambrian Ocean. The phylogenetic studies using molecular data from modern animals to reconstruct the evolutionary tree of body plans are a powerful tool for interpreting Cambrian fossils. By placing Cambrian organisms in the evolutionary tree built from molecular data of current organisms, paleontologists can make predictions about the biology of those Cambrian organisms that go beyond what fossils alone can reveal. If the molecular tree places a particular Cambrian organism as an early relative of modern arthropods, for example, we can infer that organism probably had aspects of arthropod biology not preserved in its fossils. The Cambrian faunus of regions, including what are today northern Africa, Arabia, southern Europe, India, Australia, and Antarctica, developed their own regional elements, though with the same fundamental groups. The trilobites of Gondwana were in many cases different at the species level from those of Laurentia, reflecting the geographic isolation of those tropical seas and the differentiation that began once the fundamental body plans were established. The fauna of the Siberian Cambrian, outcropping in extensive regions of northern Russia, has also produced important evidence about the global distribution of period organisms. Siberian early Cambrian trilobites show relationships with those of China and also with those of North America, reflecting the dispersal routes existing between those continental masses through the Cambrian Ocean. Understanding the global distribution of Cambrian fauna has improved enormously in recent decades thanks to international collaboration of paleontologists from all continents. The discovery of new exceptional preservation sites in Australia, Morocco, Poland, Siberia, and other regions has added geographic dimensions to a picture previously dominated by findings from the Burgess Shale and Changjiang. 540 million years. That is the time separating us from the Cambrian explosion. To put that number in perspective, it is worth comparing it with other time scales more familiar to us. Non-avian dinosaurs existed for approximately 180 million years. That means the time separating us from the Cambrian explosion is 3 times the duration of the entire age of dinosaurs. In other terms, if dinosaurs disappeared 66 million years ago, the Cambrian explosion occurred before the dinosaur extinction by an amount of time equivalent to 7 times the time that has passed since that extinction. The Cambrian also invites us to think about what it means for something to be ancient. 540 million years is ancient by any measure relevant to human experience. And yet, in the perspective of the universe, the Cambrian is not particularly ancient. The universe itself is 2.5 times older than the Cambrian explosion. The oldest stars in our galaxy are 12 times older. Even our solar system is 8 times older than the Cambrian. The Cambrian explosion, that seemingly incomprehensibly ancient event, occurred in the relatively recent cosmological past. The Cambrian also raises questions about the relationship between life and planetary processes that extend beyond biology into Earth system science. The diversification of Cambrian animals changed the planet in measurable ways. The increase in bioturbation changed ocean sediment chemistry. The proliferation of calcite-shelled organisms altered ocean calcium carbonate chemistry. The increase in biological productivity changed the ocean carbon cycle. Those biological effects on planetary chemistry were not minor perturbations. They were significant changes to the major geochemical cycles that regulate Earth's climate and habitability. From the Cambrian onward, life became a geological force in its own right, shaping the planet as much as being shaped by it. The sense of wonder that the Cambrian produces is itself a product of evolution. The cognitive capacity to be astonished by the strangeness of the ancient world, to care about what lived in an ocean 500 million years ago, to find meaning in that genealogy connecting the Cambrian to the present, is a capability that evolved in the human lineage over the last few million years. That we can be moved by the Cambrian is itself an evolutionary achievement, one that no Cambrian organism could have anticipated or understood. We carry not only the biological inheritance of the Cambrian, but also the cognitive capacity to reflect on that inheritance and to find it beautiful. The peace that comes from thinking about deep time is not the peace of indifference. It is the peace of scale. When the full scale of geological time becomes even partially imaginable, the urgencies and anxieties of daily life do not disappear, but they find their proper size. They are real and they matter, but they are small in a story that began 540 million years ago in a warm ocean full of creatures inventing eyes and shells and predation, and that will continue long after the last human thought has concluded. That perspective, that sense of participating in something so much larger than any individual life or any human generation, is what the Cambrian offers to those willing to sit with it in the dark. That reframing of deep time in cosmological perspective can produce a kind of peace. The Cambrian was not a beginning. It was a relatively recent episode in the history of a universe much older and much larger than any ocean. The organisms of the Cambrian were as recent and as temporary as we are in the context of cosmic time. And the universe that contained the Cambrian ocean still contains us, and will contain whatever comes after us, with the same indifferent continuity. The reflection on the timescale of the Cambrian also invites thinking about the future of life on Earth. If the Cambrian explosion occurred 540 million years ago and complex life has been diversifying since then, how much more time does complex life have on Earth? The Sun has approximately 5 billion years of life before its expansion as a red giant destroys the terrestrial biosphere. In that scale, complex life on Earth is still in its early youth. What evolution could produce in the next 500 million years is completely unpredictable from our current perspective, but the Cambrian reminds us that when evolution has that time available and conditions are favorable, it can produce worlds completely different from anything that can be imagined. The humility that geological time produces also has a practical dimension. The human species has been on Earth approximately 300,000 years, and its civilizations have lasted barely 5,000 years. In the context of the 540 million years that have passed since the Cambrian, those numbers are insignificant. And yet, in those 300,000 years, humans have had an impact on the biosphere that in many respects is comparable to the extinction events in the geological record. That disproportion between our age as a species and our impact as a species is perhaps the most important lesson that geological time can teach us about our relationship with the planet we inhabit. And yet, that planet with 4.5 billion years of history, that planet that has seen oceans born and die, that has seen continents form and break apart, that has seen life invent itself over and over again after each great extinction, that planet is still here, with its oceans and its moon and its drifting continents. And we are here too, for now, listening to its story. The comparison between the Cambrian timescale and other scales the human mind can handle is also useful for thinking about the nature of geological time. The universe is approximately 13,800,000,000 years old. Earth is 4,500,000,000 years old. Life on Earth is approximately 3,800,000,000 years old. Complex life, in the sense of multicellular organisms with differentiated tissues, is approximately 600 million years old. And complex life with recognizable body plans from modern groups, the life that emerged from the Cambrian Explosion, is 540 million years old. The Cambrian Explosion occurred not at the beginning of time, but when the universe was already 9 billion years old, and the planet already 4 billion.